I'd like to thank the conference organizers for the opportunity of sharing insights on the genomics of DNA repair defects in breast cancer. These are my disclosures. So my talks today will cover specific DNA repair defects in breast cancer, in particular homologous recombination deficiency or HR deficiency, mismatch repair or MMR deficiency, and base excision repair or BER deficiency. I'd also like to seed the idea of thinking about mutational signatures in alternative ways to inform patient stratification. So in this plot, we can see the breadth of driver mutations that are present in breast cancers and the relative enrichment of these different driver mutations in ER positive or ER negative cancers. And many of the genes are involved in DNA repair pathways. So historically, genomic stratification has been one where we seek the presence of a mutation in a gene in a very binary way, and then we use that to guide patient stratification. But in fact, the human genome, the human cancer genome, has many thousands of passenger mutations that may be informative because they serve as an archaeological record of the mutagenic processes that have occurred through tumor development. So this is the principle of mutational signatures. Now, we've had some knowledge accumulated recently of the sort of etiologies of several mutational signatures, but certainly not all of them. And when we sequence a cancer genome, what you see is a final portrait. It's a composite of all those signatures added together. In fact, the first report of mutational signatures was in breast cancers in 2012. And with only 21 whole genome sequenced breast cancers, we could already identify signatures of DNA repair abnormalities, specifically signatures that were enriched amongst cancers from women who had germline BRCA1 and BRCA2 mutations. By 2016, we had sequenced a larger number of breast cancers, 560 in total, and we found a larger number of signatures, 12 altogether. However, it's worthy to note that the first 5 signatures that were identified in just 21 cancers were very common signatures. They're present in many different breast cancers, and hence they were easily detectable even when the numbers of samples were low. The increase in scale and power afforded by sequencing 560 breast cancers permitted the detection of relatively infrequent or rarer signatures present in fewer samples. If we look at the causes of these signatures, so signatures 1 and 5 are associated with age of diagnosis. Signatures 2 and 13 are due to the activity of the ApoBec family of enzymes. Signatures 17 and 18 in 2016 were of uncertain etiology, and today there are causes for these that have been reported more recently. So the vast majority of mutational signatures, however, are associated with DNA repair pathway defects, including HR, MMR, and BER. So if we look at the prevalence of the mutational signatures per sample in this way, the top panel shows absolute numbers of substitution mutations, and the bottom panel shows the same information presented as proportional bar charts. The different colors represent different signatures grouped by etiology. And more than half of the cohort are characterized by the purple signatures 1 and 5 that are age associated. Actually, the majority of these patients tend to do well on standard of care therapy, and most of them are ER positive tumors. Now, just over 20% of the cohort have these HR deficiency signatures in blue, signatures 3-8. And then just under 5% of cancers have these green signatures, which are mismatch repair deficiency signatures. And then if I draw your attention to the pink signatures, which is ApoBec, a lot of samples have a small amount of ApoBec mutagenesis, but 10–15% are dominated by ApoBec proportionally. And also in terms of absolute numbers, there are some samples with marked hypermutative phenotypes. But this is quite heartening as a diagram because it makes us feel that we should be able to use mutational signatures to classify samples just by looking at this graph. There are some caveats, however. Mutational signatures don't all behave in the same manner, and some are subject to more uncertainty than others. So in this PCA plot, which is of attempts to extract mutational signatures from a breast cancer data set, we see that this signature 17, is easy to extract. No matter how many times you extract, you do an extraction, you can pretty comfortably find this signature. It forms a very distinctive cluster. And that's because it has clear characteristics with defining tall, pink T2G and T2C bars that stand out above the rest of the 96 channels. By contrast, this collection of different signatures, including signatures 3, 5, and 8, are less discretized and are more difficult to separate from one another, and they get miscalls for one another as well. And that's because they're pretty flat and featureless. So I emphasize this point because signature 3 is a feature of BRCA deficient cancers and is sometimes on its own used to define BRCA deficient tumors. But you can see here that it can be difficult to be sure that you do indeed have signature 3 in your data set, never mind in your specific tumor. And so stratifying tumors based on these individual, relatively uncertain and featureless signatures may result in erroneous tumor classification. And that is why we tend to use all of the mutational information that is present in a whole genome to complement the substitution signature. So here we have the chromosomal ideogram on the outside, and that's a substitution pattern for this particular patient. We can include indels for which you get a very particular pattern here. An excess of deletions with microhomology at the breakpoint junction, a classical pattern associated with deficiency of HR. And that's underscored by the fact that this woman has a BRCA1 germline frameshift mutation. Copy numbers next, we have a lot of pink losses throughout the genome in this patient. We can look at the structural variation or rearrangements, and here the patient has over 300 rearrangements beautifully distributed throughout the genome, and they're all of a particular class called tandem duplications. So many of these patterns that I've just described to you are the classical mutation patterns of BRCA1 deficiency, and sometimes referred to as BRCAness by the community. So here is that same patient, and here are the cancer genomes of 2 other women, neither of whom have germline BRCA1 mutations, but instantly you can see that they're indistinguishable from each other. In fact, the woman in the middle has a somatic mutation in BRCA1, and the one on the right has promoter hypermethylation of BRCA1. Each of these patients has individually thousands of mutations, not a single one shared between them yet. Taking this holistic cancer genome profiling approach, you can instantly recognize the biological state of a cancer as being BRCA1 deficient, even if how they have become BRCA deficient is different. And that's because what they do share in common are the patterns, the signatures that are pathognomonic and distinguishing. So we can apply machine learning methods to pick out these distinguishing signatures and use them to create predictors and to facilitate cancer genome interpretation. So we created something called HR detect. High scores are associated with BRCAness and low scores not. And when we've taken HR detect and applied them in the 560 whole breast cancer genomes that had been sequenced in the past, we found a higher proportion of breast cancers that had high HR detect scores than anticipated. 22% of the cohort, that is 1 in every 5 breast cancers examined, had a BRCAness phenotype. Not 1 to 5%, 1 in 5 cases. And this was, way above what we anticipated from germline BRCA1 to frequencies. And the causes for that high HR detect scores were germline and somatic changes, including epigenetic changes in BRCA1. But in a third of cases here in gray, we could not find a genetic or epigenetic cause to explain the mutational signatures for the high HR detect scores. So this is important to note because if we had taken a targeted sequencing strategy, we would detect the purple and blue categories. We'd only detect half of the orange ones because half of them are due to epigenetic modifications, and we would miss all the ones in gray. The question is, does it matter? And we think it does. For in a separate study on triple negative breast cancers, HR detect was shown to be able to prognosticate, with high HR detect scoring cases showing better outcomes on standard of care therapy. And when this was irrespective of whether we found the genetic or epigenetic cause for the signatures. So the group highlighted by the yellow arrow has been split in the Kaplan-Meier plot below into those with BRCA1/2 drivers that we could identify in black versus those that we couldn't identify a driver in gray. And as you can see, there's no difference in outcomes between those 2 lines. So HR detect was able to prognosticate even when you couldn't find a driver. In fact, the HR deficiency signatures in the form of HR detect has been shown to have early predictive capabilities as well, underscoring the value of using combined signatures of HR deficiency for clinical purposes. So I'm going to move on to another repair deficiency, which is mismatch repair. This has been observed at a relatively low frequency, 1 to 2% of the total breast cancer population. They have a high burden of substitution and indel mutations and look very different to BRCA deficient cancers. And the arrows are pointing to the indel component, which is almost a solid ring in these MMR deficient cancers. Now, in a separate and recent study of systematic CRISPR knockouts of DNA repair genes, it was shown that when MMR is compromised, tumors have much more to share, to show you than just being microsatellite unstable or having a high tumor mutational burden. There are quantitative differences in substitution and indel burdens between the different genes, and there are interesting qualitative observations. MSH2, MSH6, and MLH1 are very similar to each other and distinct from PMS2 in both substitution and in indel profiles, with more T deletions seen in the PMS2 knockouts compared to the other three. And this is reflected in human cancers as well. So here are the breast cancers caused by MLH1 mutations. I showed you this earlier. By contrast, this is a PMS2 mutant breast cancer with different substitution and indel profiles. Now, here's another mismatch repair deficient breast cancer. But what I'd like you to note is that in addition to having mismatch repair deficiency, this patient also has a large amount of rearrangements of chromosomal instability as well, driving home the message that tumors can have multiple distinctive and important signatures to detect. And here is another case to highlight another point, which is that 10 to 25% of mismatch repair deficient breast cancers are present at subclonal levels. So this is a case where a patient has heavy ApoBec-related mutagenesis. The gene MSH2 has been hit twice, resulting in late onset subclonal mismatch repair deficiency in this ER positive cancer, which didn't do well, in fact. Okay, so on to my third example of a DNA repair deficiency. Signature 30 was identified in 2016. Its etiology was unknown at that point, and soon after, the Clever's lab used CRISPR editing in an organoid to show that mutations in NTHL1, a BER DNA glycosylase, produced a signature that was identical to signature 30. They also showed that germline inherited NTHL1 mutations were associated with this signature, as demonstrated in this breast cancer, which has a hypermutative phenotype of signature 30, but interestingly has also got a BRCA deficiency. So this woman has 2 germline abnormalities, again, reinforcing this point that actually when you do a whole genome, you get all the information necessary to interpret individual cancer genomes. And I don't have time to go through all the other potential DNA repair defects, but it has been shown that there are knockouts of certain genes like OG1, EXO1, RNF168 that show similarities with signatures 18 and 5, which are also seen in breast cancers. Now, although I've emphasized sort of specific DNA repair defects, what I really want to also communicate is that there isn't just a one-to-one mapping of one signature, one cause, one therapeutic option. We need to be a little bit more nuanced about how we think about signatures for clinical benefit. And to show you an example, here are signatures of structural variation, rearrangement signatures 1, 3, and 5. These are characterized by long tandem duplications, short tandem duplications, and short deletions, respectively. Each is associated with different genetic abnormalities, but all have microhomologous sequences at the rearrangement breakpoint junctions that are characteristic of Pol theta mediated end joining activity. So these are diverse signatures with different causes, yet all may be dependent on Pol theta activity and could be used as biomarkers for Pol theta inhibitors going forward. So this is just a principle that I wanted to communicate, where the genomic readout, the consequential patterns could be due to many different or diverse etiologies, but could be used clinically in a different way to identify potential drug sensitivities. So I'm going to evolve this conceptual description of mutational signatures in cancers to add on complexity that we could exploit. Because as a cancer becomes more divergent from normal, some signatures, including relatively benign endogenous ones, could be secondarily amplified in quantity or changed in quality by other perturbations. But for translational purposes, the focus should be on identifying signatures that either report targetable abnormalities that are specific, like HR, mismatch repair, or BER deficiency, or more general biomarkers like the rearrangement signatures that I showed you earlier. I acknowledge that there are other pathological states that may also be therapeutically informative, and all of these clinically relevant signatures are highlighted in blue. Bottom line is, clinically relevant mutational signatures are the ones that will be helpful for translation. So with that, I'd like to acknowledge my lovely, highly international team for their passion and energy, even though they tried to kill me yesterday at ice skating. And I'd like to thank my funders, CRUK, NIHR Wellcome, and the Dr. Joseph Steiner Foundation, and of course, all the lovely collaborators, the patients, families, and clinicians that have contributed samples to the work described today. Thank you very much. I'm happy to take any questions. Thank you.